Gay School of Medicine, University of Southern California, where he investigated the inter- and intratumoral heterogeneity of different types of cancers using technologies such as single-cell sequencing and spatial transcriptomics. His ongoing research includes a DoD-funded study on the role of inflammation in the prostate cancer in West African and African American men, and an NCI U54 supplement funding on genomic profile of pancreatic adenocarcinoma in Nigeria. He currently the co-PI on the NCI Center of Global Health Awards that is aimed at increasing the capacity of trainees and resident doctors in cancer genomics. Dr. Rotimi is the Director of Translational Research for the Prostate Cancer Transatlantic Consortium and the African PI for the NCI Clinical Proteomic Tumor Analysis Consortium. He currently serves as the co-chair of Aortic Research Committee and Aortic Cancer Genomics Special Interest Groups. It is his hope that his studies will contribute to reducing the burden and disparity of genetic disorders among Blacks and training for the next generation of African cancer physicians and scientists. Please join me to welcome this erudite scholar, Dr. Solomon Rotimi.
Yeah, thank you very much for that beautiful introduction. And it's my pleasure to be here. Yeah, once again, I want to appreciate the organizers of this meeting for extending the invitation to speak to me. I consider it as an honor, and particularly Dr. Segun Fatumo for telling me I'll bring you and to give this talk again when he heard me speak somewhere. Okay, I know we had some technical issues, and then also after eating pounded yam, I know it can be very heavy, but I'll try as much as possible to make it, lighter for all of us. I also ate lunch myself. What am I supposed to press? Okay, yeah, just a way of background. I've been asked to speak to the role of cancer genomics in the achievement of the SDG. And the SDG that comes to mind readily is SDG 3.4. And the focus is that, the indicator that is used to measure the progress towards this SDG is the cumulative probability of dying from 4 non-communicable diseases. And one is cancer, cardiovascular disease, chronic respiratory disease, and diabetes, within, the next, 20 years, thereabout, between the age of 30 and 70 years. And so if you look at it, of all of these, cancer plays a very important role. But before I come to the specific role that cancer plays and how genomics could help us to, understand it, the question is, who really is an African? And this is a very difficult question to answer, I must tell you, but I'll give you my perspective. And I want you to also think about who you think an African is. The question is, when you hear the word Africa, does this come to your mind? Right. What about this? Where you have Sub-Saharan and North Africa? Oh, you see, yes. Okay. Sorry. Oh, okay. I think this thing is jumping too much. Okay, I think we're having an issue. Okay, go back. Okay, anyway, but however you look at African as in congregation of different countries. Okay, so the question is, what is my opinion? My opinion is that if we're talking about African, we must make effort to disaggregate Africa along the human evolutionary tree. Okay, therefore, as much as we try to compare people from East Africa with West Africa, we're going to be repeating the same error of aggregating the whole of human population together. So our efforts will gradually be driven towards disaggregating African by human evolutionary tree. And what do I mean by human evolutionary tree? You see, this tree has been well established that human population can be grouped into 5 branches, 4 of which are in Africa. And we may do very well to disaggregate our population across these 4, 3 branches. Anyway, So the question then becomes, where did cancer come from, particularly to Africans? How did we get here? Well, just to give you a background in history, that actually the first record of cancer was in Africa. It was reported by an Egyptian physician called Imhotep. And he described it thus. And you see, when it was Imhotep, it's about 3000 BC ago. Now, Imhotep described case 45, and he described it as the bulging tumors of the breast. And his prescription for treatment was simple. He said, there is no treatment. That was what Imhotep said about 5,000 years ago. The unfortunate reality of many patients in Africa today is that truly there is still no treatment. And the challenge before us to achieve the SDG is to understand and develop treatment that is suited for every human being. And moving forward, the projection has shown that by 2040, the burden of cancer is going to be in Africa. The burden is going to reduce in Europe, in America. And if you look at Africa, Africa is going to bear the highest burden. And if you divide Africa into different regions, you have the burden in East Africa, Middle Africa, and West Africa. That is where you're going to have a lot of the burden of cancer. And if you're studying, those are the populations that we need to focus our attention on the more. But progress has been made in cancer in other parts of the world. And what I'm showing you is the progress that has been made in increasing the survival rate of cancer in the United States. And you look like a cancer of the prostate, for example. The survival rate has increased from about 70% to 98.6%. That is a huge progress. How did they do it, or how are they doing it? And it's not just in the US. In South Korea, the progress has been very fantastic. In Europe, it's the same thing. Okay, so what can we learn from them, and how have they been able to achieve this? And so as I go on, I'll be asking questions of making statements that people have posed at me. You know, I told you I made a presentation somewhere, and Dr. Fatima was there, and his question I still remember was, what is wrong with Africa? And I think that would be a good way for us to start. To say, really, is there anything wrong biologically with us? Well, it wasn't the first person to ask that question. Actually, everybody in this field, after seeing the data, you will ask that question. But in 1927, this paper was published that asked the same question. The Negro, which is a black person, is he a biological inferior? And the authors clearly said that one, well, it appears to be biologically inferior because of what we're seeing. Because, for example, I just said, oh, cancer kills more people here in Africa than other places. And then he described the way we look, our nose, our eyes, and all of that. However, I said that the sweeping claims to white superiority are not justified on any present knowledge. And the more we look at it, the more we find. And around the same time, a paper was also published based on Nigerian population that showed that actually between 1900 and 1920, from the data collected by European physicians practicing in Nigeria, clearly showed us at that time cancer was very rare. That was just about 100 years ago. The question is, what has changed in the past 100 years that has made cancer to be what is so popular as it is now? And so from what we know that cancer is being described as the emperor of all maladies. But we know that that emperor sits on a throne, and the throne that it sits on is the genome. And that's why we need to understand how we can use that throne to dislodge it. So cancer, primarily, you can describe it in several ways. It's a disease in which abnormal cells divide uncontrollably and all of that. And while we're chatting, I use metabolism to describe cancer for you. But cancer, as its basic essence, is primarily a disease of the genome. And I can say that again. When a cancer patient sits before a physician, what is presented before that physician is a genome that has gone haywire. It's a genome that has gone completely crazy, if I may use that word. And we need to understand how to correct that error, how to wind back the hand of clock. And there are several things that can cause it to go haywire. There could be intrinsic factor, there could be biotic factor, there could be abiotic factor, there could be environmental factor like smoking, all of those. There are several things that could go wrong. And so in the next couple of minutes, I'll try to look at examples of the studies that others have done, as some of which we have also done, on cancer genomes, particularly in Africa, and I'll be focusing on the leading cancers in Africa. Excuse me, breast cancer and prostate specifically because of time. Breast cancer. So a physician told me, I just knew that this thing that I see in clinics is different. He said, I presented her with this data, and I will show some of the data in this place. And she said, I knew that what I was seeing is different from what's in the textbook. And the question is, what are they seeing that is different from what is in the textbook? So one person that has done a lot of work on cancer genomics in Nigeria precisely is Dr. Funmilola Pate from Chicago. And I'll just take one of the studies that she has done. And I'm sure this figure is familiar to all of us here, right? It's a GWAS study. And we're looking at what are the straight, SNPs associated with breast cancer here. And you see, they found that SNP. Eureka, yes, that's a good SNP. But the question is, and so what? What does it mean? This gene plays a part in melanin synthesis. It determines how dark we are, okay? So the question is, why should that be a problem? It doesn't make any sense, right? Well, but at the same time, evidence started emerging that vitamin D plays a role in inflammation. And because of the darkness of our skin, our ability to synthesize vitamin D is reduced. You see, and so what? And that is it. You have a SNP that makes you dark. You are designed to stay in the sun. We are here, not in the sun, wearing 10-piece suits, and that is it. There's vitamin D deficiency. It drives up inflammation. And that interpretation came from evidence from genetic studies. And several other studies have been done. But because when we're talking of cancer, we're talking of two types of genetic state. We're talking of the germline, and then we're talking about the somatic. Now, in treating cancer, what you want to focus on is in the somatic. And a lot of the study that I do is that I look at that somatic changes. And in breast cancer, another study that the Olopade team they have done is to look at the somatic mutation. And you see up there, they have the 3 subtypes of breast cancer. And no matter the subtype, the leading mutations, you have them in TP53, BRCA1, BRCA2 within the tumor itself. Okay? And when these mutations are aggregated, it results in what we call genomic instability within the tumor. Now, so remember I've talked about BRCA1, BRCA2 mutation. Now, what does this mutation also do when you have them in the tumor? And now, these studies have came out that show that when tumors carry this mutation, they respond very well to a type of drug that blocks what they call PD-1. Now, what I'm telling you is that there's when you block this PD-1, which is an immune response, material sort of, then they respond very well to a particular kind of immunotherapy called anti-PD-1. And it showed in this data that the tumors which carry this mutation respond better to anti-PD-1 than to the standard of care. And so which means in a Nigerian population that you have high mutation of BRCA gene in the tumor, there's now evidence coming out to show that this particular kind of drug will do better, or our patient will respond better to this kind of drug than the current standard of care that they receive. And so remember I talked about PD-1. That means when PD-1 is high, you block it, and that means the tumor will respond very well. The question is there evidence that PD-1 is upregulated in this population? Now, these are my students who just finished their masters, and we looked into that. And we showed that PD-1, which is the first, I don't know, sorry, apologize, the slide cut off. The first figure there showed PD-1 level in breast cancer cases versus control. Up, the PD-1 is higher in breast cancer cases in our population. And we also went on to show that we actually found a surrogate indicator that will mirror the PD-1 level as phospholipase A2. We also went to the immune system to look at myeloperoxidase. We showed that the myeloperoxidase in the cases was downregulated. We then went ahead to show what is happening with vitamin D, okay, that we showed before. Vitamin D was lower in cases than in control. And we look at a biomarker for inflammation, C-reactive protein, and we show a negative correlation between vitamin D level and inflammation. So the vitamin D level is low if C-reactive protein is high. Okay, so around the same time that paper came out and talked about the reality of cancer in Nigerian population, there was a lot of argument going on. Around the same time, this paper came out that talked about also saying that, look, there's freedom of Negro race from cancer. And if you can look at the down part that I circled, talk about northern population. So in northern Nigeria, the extreme reality of cancer may be judged by the fact that the only recorded case at any rate in recent years was one breast cancer in an old woman. And he said, well, actually, if there was breast cancer, we'll see, because the women that time, they were walking about naked. And so, if they were walking about naked, that means they were getting good exposure to sun, which is able to give them adequate vitamin D. But things have changed now, and you think about that. Let's go to prostate cancer. Let me not, let's go to prostate cancer because of time. But again, you've seen the effect of the environment and the impact that is having on our genome and how it's influencing the outcome and the risk of disease. Or just hold that thought, and let's look at what we have done, what we have in prostate cancer. A professor of urology told me, said the presentations are different from what is in the textbook. They're not typical, they are atypical presentations that come to clinic, and it's giving us a challenge. The question is, what happens with prostate cancer? Prostate is an hormone-driven, the growth of prostate itself is driven by testosterone. And the prostate cancer itself is largely hormone-driven, testosterone-driven. Indeed, this gentleman got a Nobel Prize for showing that by cutting off the source of testosterone, you can cure the cancer. That is as strong as it is, and it's a very good, it's a standard of care. However, the problem with prostate cancer is that there's a racial disparity because Black men bear the burden of the risk and death from this disease. Now the question is, what is responsible for this, the racial disparity? My PI of the PI of our consortium, Professor Odedino, and our group had shown that there is a racial disparity between testosterone level, in males, in white and black, show that, and the level is higher in blacks than white. So that's on one hand. However, we know that when you treat them with testosterone deprivation therapy, after some time, castration resistance develops. And in Nigerian population, it's been shown that about 30% patients develop castration resistance by this author within 11 months. Okay. And so the question, and people have also said that, look, maybe these cancers are there, but we're not picking it on time. Early diagnosis, is good so that the cancer can be cured. Great, absolutely correct. You catch it early, then you treat it early and you cure it. The question then becomes, how early is early? How early is that early? As to answer that question, this paper, okay, I thought you were doing that. Okay. This paper, just came out. They looked at prostate in autopsy cases in Nigeria. Indeed, the summary of it is that it's not as high as expected. And so what it means is that it's not that these cancers grow and they are not caught early, and therefore they outgrow us. The issue seems to be that the rate of growth is very high, and we call that aggression. The question that we are asking, therefore, is that what drives prostate aggression in Black men? And so one of the studies that we've done, particularly when you look at testosterone pathway, is that to look at CYP3A4. I put this because of the conversation yesterday. That CYP3A4, what it does is that it oxidatively removes testosterone. So if you are able to remove it early, then it will not continue to stimulate the growth of the tumor. And so there's a particular SNP that is associated with reduction in the activity of that protein. And that's CYP3A4-1B. And that SNP has been shown to be associated with prostate cancer in African American. And we tested it in Nigerian population, particularly Yoruba population. We analyzed 150 cases versus 150 control. Ladies and gentlemen, everybody carried the same mutant gene. Everybody. Okay? Cases, control. There was not a single exception. And we said, therefore, this allele cannot be a risk factor for prostate cancer in Black population. Rather, it's a surrogate indicator of race. It's a surrogate indicator of ancestry. It's a surrogate indicator to show that you are from this part of the world. Okay? And there could be other things that will expose you to that risk. And so we started thinking, what else does this gene do? Oh, good enough. It's associated with vitamin D. Vitamin D signals and controls the upregulation of this gene. You want more of this gene to function very well, you need to have adequate vitamin D. And then that led us back to our vitamin D pathway. I started studying vitamin D, and I had this MSc at that time, and we look at vitamin D receptor polymorphism and vitamin D binding protein. And we show that the mutation in vitamin D binding protein is more significant, it's more important than the mutation in the receptor. So you must be able to also transport the vitamin D. But at that time, we didn't quantify vitamin D level. But also, thereafter, we got this funding to look at the involvement of vitamin D in inflammatory pathway in prostate cancer in Black men. We're looking at African American and Nigerian population, and of course some people from Ghana. And my PhD student that who recently I think Senate approved his PhD about the last Senate meeting 2 or 3 weeks ago. And so Dr. Adilani, I called him now, and he was at Tuskegee and at Campbell's lab at Ohio State University. And we're looking at what does vitamin D do to prostate cancer, genetic signature. And we found that was that I will summarize is that you see this immunohistochemistry and this immunohistochemistry, and I understand majority may not understand what this is, may not be familiar with it, pardon me, is that we look at the tumor, instead of doing RNA-seq, we look at a stain that we look for that X protein itself and we stain it in the tumor. And this is very useful for pathologists to know the localization of that. And we show that the genes that are co-regulated, that we consider as co-regulators of vitamin D, SMARCA1, BAS1A1, are also downregulated. And we show that vitamin D, actually Clayton has shown before that time, so I take that back. Dr. Clayton yet already showed that vitamin D receptor is actually a transcriptional factor that requires vitamin D to be activated. So when you have adequate vitamin D receptor is activated and it does all the good job it should do. You have deficient vitamin D receptor is not able to be properly activated, and then things go awry. And what is the vitamin D level in prostate cancer in Nigerian men? We recently got this data finished about a month ago, and we show statistically significant low level of vitamin D in prostate cancer patients, lower than colorectal cancer in Nigeria. And so with the evidence of this study, and of course, relying on, this disappeared, I will explain this. Relying on evidence of this, also this additional study by Adina, that actually, when men, and what they did was that when men presented with prostate cancer, they take the first biopsy for diagnosis. They did RNA-seq. They placed them on vitamin D for 2 months, and on second biopsy, they did, another RNA-seq. And they showed that the first, RNA-seq for the, pre-diagnosis biopsy, you could clearly separate the tumor of Blacks from white men, and there's high upregulation of inflammatory gene in the prostate tumor of Black men. However, after 2 months of supplementation of vitamin D, the inflammatory signature is almost extremely reduced. Okay, so you are able to use vitamin D administration to remove, to bring down the level of that inflammation. And we are now about to do a clinical trial in Nigeria that will be now administering 2 months vitamin D to prostate cancer and study the immunoinflammatory response. And so beyond vitamin D, we have also done, whole exome sequencing. And from our whole exome sequencing, we also see upregulation of BRCA mutation, high level of BRCA mutation in the prostate tumor from Nigeria, which is higher than African Americans and European men. And we know that when there is BRCA mutation, such tumors respond to PARP inhibitors very well. Okay, yeah, I'll go through. And evidence that has shown from clinical trial to show that PARP inhibitors, patients treated with PARP inhibitors respond better than standard of care, particularly when they carry BRCA mutation or mutation in BRCA1, BRCA2, or ATM. And so that's the approval. Now, as I begin to round up, I just wanted to also show you that all the studies I've been talking about, whether you talk about, whole genome sequencing, whole exome sequencing, these are what we call box sequencing. But we know that cancer itself, the tumor itself is highly heterogeneous. It's made up of diverse tumors. That's why you have cases where you say a patient has been treated, it does very well, it goes back home, and after some time, the tumor regrows. The question is what is happening because of one of the factors is because there's something we call tumor heterogeneity, in that not all the population of the cells behave the same way. And there are now genomic technologies that can be used to understand this. One of such is that, so when you do, box sequencing, what you do is that it's as if you are preparing a smoothie, you blend all the DNA together, but when you do single cell sequencing, it's like you are preparing a plate, of fruit salad. You are able to individually identify each tumor and know the genetic, the DNA sequence for each cell in the tumor. And of course, the technology, one of the earliest technologies that came out that we used during my Fulbright fellowship was the one developed, by what's the name of this company now? The name escaped me for whatever reason. But then it relied on, picking the tumor cells one by one, and using a technique similar to droplet PCR, prepare your library prep per cell and sequencing it. And of course, it goes to Illumina. And this is the kind of thing you see that come out after your bioinformatics analysis, in that you have this here, you have about 490 divided by 2, that's about 245, or so cells. And each column that you see represents chromosome, and each line represents cell. And what you begin to see is that you see that the amplification you see varies from one cell to another. And you have a subpopulation of the cell that carry unique amplification, particularly on chromosome 3. So which means if that amplification poses for drug resistance, the drug will all the other cells will respond to that drug, but this particular subpopulation of the cell will not respond to that drug. Okay? And as soon as you remove the evolutionary pressure or the survival pressure, this other subpopulation begins to grow again. And so we now have genomic technology to understand this type of variation, intertumor variation or heterogeneity. And finally, I also just wanted to touch on spatial transcriptomics and to show you what it looks like. So think about it that you take this tumor, you take your cell, you take your biopsy and you homogenize it. Or whether you want to do single cell sequencing, you still have to dissociate it to individual cells. The problem with that is that we lose what we call the spatial construct. You do not know where these particular unique cells are located in 2D. There's technology now, which is called spatial transcriptomics, that could be used to understand this. And I think for the sake of time, I'll just gloss over that explanation. And just a concluding thought that cancer is a disease of the genome and not a disease of the tissue. And so for us to fully understand it, we need to begin to move towards what we call tissue agnostic therapy, which is based on the genomic configuration of the tumor rather than the site of the tumor. And that means we need to upscale genomic capacity in Africa. And from our study, we have clear evidence to show that vitamin C plays critical role in immunoregulation of carcinogenesis in Black population, including Africa. And we're optimistic that PARP inhibitors will be very useful. And more aggressive and concerted efforts, of course, is needed. And that's why we are here to increase genomic studies in African population. Thank you very much.
Thank you so much, Prof. I think he deserves another round of applause. He just made the presentation to a layman level that everybody will understand. Thank you so much. But I don't know whether we have questions for him. Okay, someone here?
it's not my thing. But do you get me? So we have okay. I don't think this complex we are experiencing is linked to being educated. I just think it's a cultural something. So what do we look advise? And I'm very, very sure that even if an African go on vacation, they will not go to town. Do you think we should add it to our kind of information like the public health people used to do it, that guys, once in a week, go to your balcony, tan? That's my question.
Yeah, to the organizer, did you record his voice? Because that's very good for, publicity for, So you answer the question, okay? That is it. We change our lifestyle, we get exposed to sun. We should tan more than the white folks. Yes. The sun you are running away from is your friend. Yes. Yeah, good question. So rather than, I wouldn't want to encourage that, because you see there's a particular UV wavelength that's suited for vitamin D synthesis. And yes, there are tanning beds, there are things like that people are marketing. But you see, of course, entrepreneurs will always come up with things, but you see, the sun is free. Just as you said, once a week on your veranda, your balcony, just get exposed to it. Anywhere in the jungle, so long you can get some.
Thank you very much, sir, for that presentation. I have some comments and questions for you. On a light note, I saw, you said something like your research is to address SDG 3.4. That sounds strange to me. You mean SDG 3 and also SDG 4? That's the question. Then secondly, in your presentation, you gave us a projection that by year 2040, you talked about the cancer profile in some nations. What was the study based on?
Oh, okay.
I'm not done, sir. Then you also said that cancer was first reported in Africa, specifically in Egypt. Are you saying that cancer originated from Africa? Then the last question is this. You talked about one of the interventions for curing cancer, which is a surgical intervention that's cutting that's castration. What about, my PhD thesis was on the use of hormones in fish. I realized that farmers also use hormones, in their, production, say beef, fish, and what have you. So after castration, what about also exposing humans to, sources from maybe fish or beef? What will happen after that? Thank you very much.
Yeah, thank you. So let me take it from the back so that if I forget the one or two, you can remind me. Yeah, of course, environmental exposure to hormone is a major issue. There are folks who have actually linked it to increase in cancer as well. To say, the poultry that, beef that we eat, all of these things are, injected with a lot of growth hormone. And so they are driving what they call organic, food. And so, yes, there are evidences to show that those things are linked to it. In terms of the data that I showed, it's actually GLOBOCAN data, GLOBOCAN. It's a body that aggregates cancer incidents and risk, cancer incidents across the world, and also report mortality rate. And so based on their data, they usually project, say by 2020, but by 2040, this is what will happen. So cancer in the future, yes. It's available online. You can just go and look at it. For all the countries, for all the regions, they have the data. What's the other one? Oh, the origin. Okay, right. Oh, yeah. So again, you look at the most ancient culture in terms of writing and things like that. And so that's perhaps why it is. Again, I mean, there's evidence to show that humans came out of Africa. So you shouldn't be surprised that most diseases that are still with humans too, we have some footprint in Africa. So I'm not saying that Africa is the origin of cancer, but then I just said the oldest record of cancer. So rather we just say that the Egyptians kept a lot of records. That does not mean that it probably didn't occur in other places. I know there's one more other question. Oh, SDG, is SDG 3, and then they have 0.1, 0.2, 0.3, 0.4. Yes, that's it, yes. Sub, yes, sub. So not SDG, have SDG 3, and then there is sub. 1, 2, 3, sub 1, sub 2.
Thank you very much, the Prof. Really appreciate your presentation. I just want to ask this question because I know you have wide knowledge on cancer. You see, we believe from our biological sciences background that cancers result as a result of uncontrolling growth of the cells. But in cell communications, all the factors that are responsible, that are involved before the cells can just adhere together and differentiate, vitamin D is not there. But you inform us today that vitamin D is a factor, and I believe that's one of being here. But now you say you are going for the clinical trial. Can we now starting reporting it maybe when we are discussing the issue with students that, oh, we can involve vitamin D as well? That is number one. Number two is that you mentioned castrations resistance. I do not understand that one. Then the other thing that I want to ask again is that my little knowledge about the tumor is that if you have tumor and earlier you remove it, they will not grow. It will not grow again. But if you have tumor and then you remove surgically, probably if it remains small, it will grow. I have a little experience. I have done some surgery with canine. You understand? So we have what we call TFT, tumor feneral transmissible. If you just correct it surgically, if it just remains small, it will grow. But if you are able to remove everything, the small will not grow again. So normally what I used to do at times that I use burning. I don't know where it is not being experiment somehow, because maybe when I continue my study, it's not in that area, so I didn't have the opportunity. When we burn the cells, it will not grow. Then another thing that I want to ask you is that why some drugs are so toxic? Because I find out that the patient die as a result of toxicity of the drug earlier than the cell itself killing the patient. Please, can you just share some light on this?
Thank you. So let me start from the last one as usual. I think we should be very careful when we say drug toxicity kill the patient, because the drug toxicity cannot just kill patient. You get what I mean? Because the issue with that is that it passes wrong information about chemotherapy. So why are chemotherapies toxic? They have to be toxic because they want to kill the tumor. They target rapidly dividing cells. Okay, that's the primary thing that they do. Okay? And so when you say patient, you don't know, the patient may also have other comorbidity. And so it's difficult to just say, oh, yes, there's chloroquine toxicity. There is no drug that doesn't have adverse effect. Okay, and so I think we should just look at it in that area, in that perspective. So if you do surgical resection, right? What they do in cancer is that they radiate. They also go for radiotherapy. Sometimes they radiate the margin of the tumor or the tumor bed. Okay, they radiate the environment, which is like, so they can't use burning for human being the way you use for animal. But the question is, you are looking at the tumor from physical perspective or from histological perspective, angle. Tumor is primarily a genetic issue. And so you can have a cell that is genetically tumor, but is morphologically normal. And so when you say if you remove all the tumor, the question is, how do you know that you have removed all the tumor? That's number one. Number two, how do you know that even when you have removed the tumor has not metastasized? And so that brings us to the new technology we talk about, liquid biopsy. You say, are we able to collect the blood sample and begin to fish out using genomic technology the circulating tumor cells? Okay, so it's difficult to really know whether to decide whether a tumor is absolutely still organ confined. So that's the tricky part of it. Yeah, it can look benign, but it's already malignant. So what's the other thing, sir? Oh, you do castration resistant. Oh, simple. You subject the individual to castration, and then the tumor responds to that castration. Just hold on, sir. The tumor responds to castration. At that time, we call it castration-sensitive tumor. But after some time, the tumor can begin to grow again after the castration. So the tumor that grows after castration is what is referred to as castration resistant. That is no longer sensitive to the castration. Yes. Oh, okay. So you don't see, so vitamin D is, as I said, activates vitamin D receptor, which itself is a transcriptional factor. So vitamin D is not in our, real biomarkers that we use for cell control or cell growth. So no, it's not. It doesn't play a direct role in there.
Sorry, good afternoon, sir. Please, like going back to what he said about this vitamin D, we're talking about a supplement. But have you put it into consideration that this vitamin D also enables the absorption of calcium in the system?
Yes, sir.
And what if you have too much of calcium in the system, hypercalcemia? How do you deal with that also?
Again, so we're supplementing, vitamin D deficient people. Okay, so this person is deficient already. So you need to bring it up to normal level, to 25 millimolar per milliliter or something like that. So it's not just that people, for you to, what we are using supplementation for is to bring to the normal level. Okay, yeah, so we know hypercalcemia is a major problem.
Thank you very much, sir. Thank you for the presentation. Okay, going back to the vitamin D and tanning that you talked about, have you considered the risk of melanoma?
Oh, good question. Well, if you look at the risk of the cancers in Africa, melanoma is not.
Have you considered maybe it's because we've not been tanning? We've not been tanning like the white people.
No,
we actually, I get
you see, the problem of melanoma is primarily not the sun.
Not the sun.
It's not. You see, it's not the sun. You see, melanoma is a complex disease. So it's not, the sun may trigger it, but the sun is not the cause. There are several factors involved.
So
wrong diet, all of those things, and also low level of melanin. Melanin is an antioxidant that helps us to regulate the negative effect of sun exposure. Okay? And so, actually, if you look at the incidence of melanoma, it's common more in the northern and southern hemisphere. It's very rare along the equator.
Thank you very much.
So let me just take it. So I'll come and see that. Very brief.
Yeah. Thank you, Prof, for that exclusive presentation. Yeah, so my question is, since you said you'll be going to RCT, you'll be doing randomized clinical trial to examine the association between vitamin D and susceptibility to prostate cancer. Yeah. So there is this study, this method called Mendelian randomization, which you can actually use in lieu of RCT, just to have an idea. So there are diverse Mendelian randomization analyses that have shown that there are no association using genetic process between vitamin D and prostate cancer, even with breast cancer. One of them was one from Professor Lopade. So they didn't really see association between, they didn't see association between breast cancer, vitamin D
so
and breast cancer. So
we're not doing as vitamin D and at risk. We're doing vitamin D on clinical outcome of folks who already have the disease. We're saying, does vitamin D improve their outcome? That's what we want to. You see, so the data we showed, the Olopade study and so many other studies show that, really show that, look, it's, whether you have low or high vitamin D does not really, it's not really significant enough, I said. But we're not looking at risk. We're saying these are people who already developed the disease. We know that vitamin D will improve their immune system. We're just saying, does that contribute to their outcome? Does that make them to do better? You get what I mean? That's what we are doing. Okay, let me come and see.
Sorry, Sam. Okay. Okay.
Your risk. No, okay. So I will answer you. You want me to explain anti-PD-1 pathway? That will take us come and register for my immunochemistry class. Talk to Dr. Ishewon. Dr. Ishewon, we arrange that with you, but I'll come and see you. Let's just take.
Yes, sir. I have a question, sir. Hello, sir. Can you hear me?
No, can I come and see you so that we take online?
Okay.
Yeah, we can chat. So let's talk online.
Please, we have some questions online. And before that one, I think I have a little question. Vitamin D, as I learned from chemistry that biochemistry, that it has to be early morning sun. So this tanning, I think it should be specified. The sun we actually take.
Okay, so it's actually early morning, correct, but it goes beyond early morning. So I'll send a newspaper article that I wrote to Dr. Ishewon. She will put on a platform everybody will see. There's actually an app that you can use on your iPhone to tell you the best time to get vitamin D where you are in the world. So I don't collect any money from them, so that's why.
So I have some questions. The first one, does Africa have the facility to use a genome approach to solving cancer based on your study? And the two, so are you able to reduce prostate and breast cancer prevalence in the African community to deficiency in vitamin D? I'm wondering if it's simple, what you would say about genetic predisposition and other random mutations that haven't been accounted for.
Oh, the facility. Of course, we've been talking since yesterday. The cancer genomics does not require any special facility apart from what other genomic studies require. So since yesterday, History Africa, other things, those of us at Covenant University, we should be buying our sequencer very soon. I'm sure 54 Gene has the facility. There are facilities coming up. The NCDCD person talked about, they have the facility. So it doesn't, there's no facility that's for cancer genomics. It's the same facility, Okay. Now the other one, You see, there are many. You see, cancer is a multifactorial disease. I just gave one. I just identify one. My presentation just focused on one. If we're going to talk about 10, we won't, you know, there's no time. We just say, look, what we are finding from our study is that we know, first of all, we know that inflammation has now been added to the hallmark of cancer. In Black people, what have we seen that is a contributing factor to increase in inflammation is vitamin D. We've seen that vitamin D vitamin D deficiency contributes to increase in inflammation. And there's evidence that when you administer vitamin D, it reduces that inflammation. And we're saying that that would be beneficial to cancer outcome. Oh, okay, no. So multi-drug resistance. So vitamin D is not a drug that is targeting the cancer pathway as it were. So it's targeting vitamin D receptor. And so there is no way, we're just saying what you should get adequately. Okay, it's like people having scurvy and we say take vitamin C. So we don't expect vitamin C resistant scurvy to develop. Oh, no, okay. So there are studies that have shown that multivitamin administration is not been shown to be effective. So that's why we're not talking of, our study is not focused on multivitamin. Again, I will explain. We've shown that vitamin D controls inflammation. Okay? And inflammation is a major hallmark of cancer. Inflammation drives cancer aggression. When you control inflammation, it improves the clinical outcome of the disease. Okay? And so because vitamin D is deficient in our population, we're saying replacing that vitamin D should reduce the inflammation, which should consequently improve the clinical outcome of the disease. So we're not using vitamin D as a targeted therapy. No.
Thank you so much. Please, any other questions should be directed to him. Thank you, Prof.
Sorry, I want to crave your indulgence. He deserves another round of applause because it was a brilliant and loaded presentation. And we thank you, Dr. Solomon Rotimi, for that. And right now we'll be having an invited speaker who will be presenting online. And our speaker is Dr. Tinashe Chikowore. Do we have him online? Is he ready for us?
Yes, I'm ready.
Tinashe? Hello,
yes,
is he ready?
Yes, I'm ready. I'm
okay,
online.
studio, please. Pick
Yes,
up Dr. Tinashe for us. Thank you very much. You have 25 minutes for your presentation.
Right, thank you.
Dr. Tinashe, are you set?
Yes, I'm set.
We're ready for you, but we can't hear you.
I can hear you.
All right, thank you very much.
I can hear you. Am I now audible?
Yes, we can hear you.
Okay. No, thank you so much, Che, for the kind introduction, and welcome everyone to this prestigious and memorable conference that we are having, the second one of its kind, as we are setting pace to understand more our genomics and leveraging also on the expertise and that we have on the continent. So delighted and grateful for the previous speakers who have come before me, and I'm now eager to take you through this talk. So my talk is entitled Leveraging Bioinformatics and Genomics for the Attainment of Sustainable Development Goals in Africa. So my name is Dr. Chinashe Kore, and I'm currently based at the University of Witwatersrand in South Africa, where I'm a Wellcome Trust training fellow there. Right, so just for the introduction, we can actually see that there's a growing interest with regards to genetics. For instance, recently around about 2018, you could see even news writers writing a lot about genetics on the global scale. For instance, a Harvard group showed that when you design a polygenic risk score, you can actually predict people who actually develop coronary artery disease. And this was also, the prediction was similar to what you see in monogenic, when you use monogenic SNPs or monogenic variants to do the prediction. And this became a news everywhere, where people were saying, no, a Harvard scientist think he has a gene test for heart risk and he wants to give it away for free. We know research is published, it's made available, but everyone was just fascinated that really you can actually do such things with genetics. Another finding, another writer wrote, just writing about this, he says they're actually like clues about your health that are hidden in 6.6 million spots in your DNA. And another one, another writer even wrote to say, this new test has the ability of actually detecting or predicting something before you even have symptoms. With all this fascination with regards to the news writers, a lot was also being tweeted. Also, when we look even in reviews that were being written, people are now saying we are now at the cusp, really, of translating, moving our genetics from the lab and bringing it actually into the clinic. So this was as a result of the findings and the promise that polygenic risk scores were actually giving to the academic community. And just to give you an example, these polygenic risk scores enable us to stratify people according to their risk. So there, when each individual has this score, which is just a single value, we are able to plot them and they form a normal distribution. And with this normal distribution, we are able to see people at high risk and those at low risk. So for instance, a clinician might be interested to see which patients should I give a more intensive therapy. So when they use this polygenic risk score, they're able to categorize people according to risk, and they'll see that those people who are even have higher risk, some of them, they would have a 3.1-fold higher of actually experiencing a microvascular or a cardiovascular event. And some of them have a 2.4-fold higher probability or risk for them developing or worsening with nephropathy. So when they've stratified people, so people actually did this, and when they stratified people and then went about to do a clinical trial and actually randomize people based on their, not randomized, but distribute people into the arms based on low risk and high risk, they then found that this intensive intervention was only beneficial to those at high risk, but not even beneficial to those of low risk. So with all this ability, that we see the promise that genetics is giving us, that finally we can now stratify people based on risk. These are things also that is making even African scientists on the African continent very much interested to say, how can we also take hold of this global space? But nonetheless, there's something that is very much worrying. So with all this good news coming from this polygenic risk scores, it actually has been found that because a lot of studies has been done in Europeans, these polygenic risk scores predict 4.5-fold less in Africans compared to what they do in Europeans, to the fact that there's now a growing concern that all these advances might actually pass people of our African ancestry. So, and further even exacerbate the health disparities. So why this all is because there's still limited or fewer studies in GWAS. So you can see here on the global scale, these are just the populations in billions. And you can see that the Europeans or people of European ancestry actually occupy a smaller proportion of the global population, but a majority of the GWAS, actually 96% has only been done in people of European ancestry. And this therefore limits the portability or the transferability. So we cannot say something which is found in Europe, no, it will really work in Africa. That might actually be a different dimension. So all this now is sort of dampening now the hopes to say, so how can we leverage on these findings, as it seems we are still way behind in terms of participants or numbers that actually have participated or have been used to come up with all these genome-wide association findings. But nonetheless, the African story is not of despair and gloom. I just want to focus a little bit on also some novel things that have been found in Africans. So there is a gene called PCSK9. So this gene actually has some alleles which makes people have very low LDL cholesterol levels. And some of the variants are only found in people of African ancestry. They're actually not found even in Europeans or even Asians. They're actually only found in individuals of European ancestry. So these variants, for instance, if you look there on the shape plot just below there where I'm circling there, so these variants, especially these two, Y142X and C679X. So it so happened that when researchers were actually looking into this, so it was in the ARIC study, so they actually just took the LDL levels, plotted them in a normal distribution curve, and just looked at individuals which have very low LDL cholesterol levels. And they found they had actually these mutations that I've mentioned. And what was more fascinating was there was a lady who actually had two mutations, like she was a homozygous mutant for all these two variants, and she had no detectable LDL cholesterol levels. So how this works is the PCSK9 is an enzyme, so it recycles your LDR receptors. So your LDR receptors are the ones that where your LDL cholesterol binds in and it is taken in the liver. So the PCSK9 role is to work on that receptor. So if you have more PCSK9, you actually have fewer LDLR receptors. So therefore it means you have more cholesterol building up in your bloodstream. But if you don't have, or if you even have fewer PCSK9 molecules, it means your LDLR receptors is not recycled, and it will always be there to take in more and more and more LDL cholesterol. So this is what they actually found, and they used a lot of African American studies, and actually seen this protection was for a lifetime. And if you compare the trials that have been done for statin drugs, they've only have been done for 5 years, but they don't even lower or their protection with regards to coronary artery heart disease. It's not as high as this, because these ones are actually having 80% protection, that if you have them, you are protected 80% throughout your life. So with this finding, it was then deemed or dubbed a gene of rare effects, to the point that drugs were created which mimic this work. And also we are fortunate to work on this just even to see, because these studies have been done in African Americans. And we are also fortunate to actually see that even in South Africa, from even as adolescents, you could actually see that they're very low LDL cholesterol levels, and they even have very low fasting glucose levels. This is very interesting, because if you look with the current drugs, which are the PCSK9 drugs, the statin drugs, they even have an issue. So, but our work is also substantiated or validated with what we had been seeing in African Americans. So you could see in these variants, the A443T, the E67G. So this is in African Americans. Then you only get one variant, which is R46L in people of European ancestry. But it doesn't lower cholesterol as does these variants that are found in African Americans. And as it lowers cholesterol like that, you can see it actually as an advantage, because when you come to the statin drugs or the current drugs that are prescribed for people who have elevated cholesterol levels, they've actually been shown also to increase your risk. So if you look here on the y-axis, that's a cumulative risk of type 2 diabetes. So the more you increase, like the dose, if you want to lower the LDL level and you increase the dosage, you are actually increasing the risk of that individual actually becoming type 2 diabetic. And as we have shown in these studies, we have actually shown that these variants they lower cholesterol, but they don't increase even your fasting glucose. They also lower it the same. So making it a blockbuster drug that really is of great interest. So as we see that, also clinical trials were actually done on the drugs that really mimic this effect. And they actually see that they have no effect on diabetes risk, as we see in the statin drugs, which when you increase the dose, you actually increase also your risk of being diabetic. So with all this, I mean, you would really see something that's interesting, that these are variants or mutations that are only found in people of African ancestry. But when you mimic their action, they have a benefit to everyone, regardless whether they're Asian, regardless whether they are European. And these drugs really are the in thing nowadays, as they have a stronger effect, I mean, in helping people to lower LDL cholesterol. So as we had started initially to say, there are studies that are happening in the European setting, but if you want to translate them into Africa, you can't. I mean, because of the great genetic diversity between Europeans and people of African ancestry. And when you even get into Africa, as we're going to see later, there's also greater genetic diversity. But what's interesting here is you can actually take a finding from an African, from Africans, and you copy or you mimic the action of these variants, and you get something that really benefits the whole world. So what it really is indicative is Africa is really a treasure trove of genetic discoveries that we can utilize to really change and help us attain these sustainable development goals. So we then set about to do a review, particularly with regards to the non-communicable diseases. Yes, indeed, we are so fascinated about the COVID pandemic, but infectious diseases, once they are contained, they can really be managed better. But there's this silent, or there's this big elephant in the room, which is actually non-communicable disease. So we're not interested just to take a snapshot what has been done on the African continent. So we first started to look at candidate gene studies. This is where studies where people just look at fewer genes or fewer genes of interest. And we actually realized that on the African continent, a lot of research was actually being done in Tunisia. The next one is South Africa. But other countries such as Ghana or even Algeria, Burkina Faso, and even Uganda, they didn't even have much activity there. So implying that a lot of research on the African continent was only being driven from Tunisia and South Africa, and even Nigeria didn't even have all this. So we are looking just at the candidate genome studies that have been published between 2009 and 2019, and then comparing the numbers. So your y-axis is the number of these studies that have been done, and you can see the majority is from South Africa and Tunisia. Then we also were keen to see how has this transgressed, what has changed on the African landscape. So people then moved, as we're looking between 2009 and 2019, so people moved from candidate gene studies, where it's just a fewer markers. Then they went into the pre-early GWAS, where they were actually using some customized made chips which have fewer variants. And these studies are also not so many. But then came the H3 Africa, which I'm also going to show later. And this really was a game changer on the African continent, as we saw biobanks being set up, computing cluster, and even for the very first time, Afrocentric arrays were actually designed. And we have cohorts of participants that are now reaching hundreds of thousands. Then something which is unique also happened, whereby there was a government and a public-private partnership, whereby the government partnered with academia and actually poured money in what you call the South African Human Genome Program, which then was able to elucidate more of the genetic variation that we see in the southern part of Africa. Then recently now, we are now in the era whereby we are now reaping the fruits from the investments from the H3 Africa, whereby people now are doing geo studies, which include 30 million variants. So if all this goes well, it actually puts Africa on that landscape of being able now to discover more and more variants that might not only benefit Africa, but even might also benefit the world. And we are also, there's also something that's also starting up, which is also key and novel, that we see that's starting in Nigeria. I mean, and it's also good that we are having this Congress where we see 54 Gene partnering with academia and also establishing the heritage study there, which is anticipating to recruit hundreds of participants. So we can see that the light is getting brighter on the African landscape as we are moving and moving and moving. And we just need, to continue on this trajectory such that we can actually therefore get discoveries that can benefit even the world and even Africa. And they still need even of prioritizing genetics in Africa, as we are actually going to see that there are even countries on the African continent that do not even have genetic studies that have been documented or do not even have an existing genetic study. For instance, in Angola, in Gabon, in Central African Republic, South Sudan, Chad, Somalia, they do not have like active genetic projects that are running, focusing especially on non-communicable diseases. And other countries, I think Nigeria is just fortunate enough that we have the H3 Africa, we have the heritage study there. We also have a cancer project that is also there, that is leveraged, and many others. But it's one of the fewer countries that actually have a lot of long-term or bigger genetic studies that are focusing on NCDs. Other countries just only have a single, and even others do not have much. And you can see South Africa there dominating with many other things. So what's even key, if we are going to attain or leverage genomics and bioinformatics, it is imperative that we get more African players to actually contribute to this work and also be part of this genomic revolution that's happening on the African landscape. As we know, African genomes are actually a rich treasure trove that can benefit many. So just to look more deeply into the H3 Africa, which is an example, a flagship project that is actually ignited a genomic revolution on the African landscape. So the H3 Africa has actually recruited 100,000 participants, 445 students or capacity development, and we can actually see they've even contributed to 373 publications, just showing the value and the utility of this consortium. And that's a picture of the meetings that are usually held. So that was the last picture that was taken in 2019, where people met physically, to discuss, about the African research. So we can see, based on what we saw in that review, the schema, that really Africa is on that trajectory, really, to getting there, to really now start to spare out genetic findings that can change the landscape. But there's also a key finding that really came. So this is just an example of a great work that came from the H3 Africa. So late last year, there was actually a big landmark paper that was published, which was based on the whole genomes of people of Africa. And they actually found that there were 3 million novel variants found in Africa, which had never, which were not on any publicly available database, thereby showing that really Africa is really that treasure trove for genomic discovery. And all these whole genomes came from all these countries. We can see we have a representation of a lot from West Africa, we have your Central Africa, East Africa, and also South Africa contributed to this great work. And this was actually published in a very high impact journal, just showing the impact that African genetics has. And we now hope that, people will leverage from this work really to even help us understand better and even come with potential treatments or even potential variants that are of clinical utility. But nonetheless, as I indicated, there are still a few players in this graph. Even our studies, they're still in smaller numbers. We're just playing in the hundreds of thousands, but we really need now to be looking at how others are doing, because others are now stretching into millions. But we are still playing a little bit fiddle behind. So what do we need? So what we need here is to really have that key perspective where we really prioritize genomics and bioinformatics on the Africa landscape. The advantage that we have, as has been indicated, Africa is going to be a burden of many of those diseases. Africa is going to be that epicenter. But the question, do we have a will to tap into the genomics to help us understand the burden of these diseases that we actually are going to be facing? Then the other pillar, which is also a short change always on the African continent, is the lack of resources or infrastructure. And it's quite key that even as our leading researchers are building up their careers, that they should also focus on getting grants that leverage or enable infrastructure to be set up. And even governments need also to play a strong role here. For instance, I gave that example where the South African government poured money just for people to understand the whole genomes in South Africa. And this is quite a good initiative. So it's also key for it might even require to rededicate infrastructure. We know a lot has been invested with regards to HIV, but this can also be rededicated or even reprioritized to ensure that even genomics can leverage on that. There's also a key for need for strategic funding. We see what 54Gene is doing there in Nigeria, but it'll also be good even if government gets involved, even other big pharma companies get involved, or even technical giants, be it Facebook and the others can also get into this space, like what we saw which happened for H3 Africa, whereby it was really a strategic funding from the NIH and Wellcome Trust that really became this game changer. And really capacity building, that is where our sweet spot is. We can have all this good genomics, or African genomes can actually be a good treasure trove, but we can still be subject to this helicopter research, whereby we are just custodians of samples, but we are not even the ones that are even analyzing or even churning out discoveries from the genomics that we really have. So it's also key for us to really focus on the emerging, the young generation that is coming, such that they can actually be well-trained. And this involves even utilizing exchange programs, even early career funding facilities that can make this a reality. But nonetheless, we need to remember that African genomics is not only of value to Africans, but it's also even valuable to the global populations. So it's also key that when we have done what we have done, we also collaborate with the global consortia so that our findings can really, really enable us to make a strong advancement, and also to enable us to realize our sustainable development goals. So I would like to thank you all for listening, and I hope I've motivated you that we really need to leverage on bioinformatics and genomics to enable us to attain our sustainable development goals. I also have to take this time to acknowledge my funders and sponsors, the H3 Africa, AWIGEN, the WITS University, my sponsors, collaborators, and also members of the African Computational Research Group. Thank you.
Thank you very much, Dr. Tinashi, for that brilliant presentation. And I was just trying to come up and you were just right on time. Thank you very much. So do we have any questions from the floor? Any questions? Any questions? You're still trying to think about it. There's no online question. So do we ask him to go? It may be a comment or whatever, just let's hear you out. Thank you very much.
Yeah, thank you. Yeah, just to appreciate him, for clearly pointing out, and he reiterated that very well a couple of times, that the reason for doing genomics research in Africa, pardon me, I was going to say it's not just for Africa, it's actually for the world. There is no way we're going to achieve equity in Earth globally without understanding the genomics of Africa. And it's so important because Africa is the bedrock, is the root of humanity. It's like you're studying, so studying other populations is like you're studying the branches. And we're saying, and I think that's a very good point that we need to begin to add to our marketing strategy, if I may put it that way. Okay, to tell people, look, when we say fund, when we say fund, when we are resourcing for funding for genomic studies in Africa, it's good for us not to just make it that, look, this is to help Africa. No, it's for the world. Yeah. We still know where you came from. That's what we are saying. Know where you came from. Know the genomics of your ancestors. That's what we're saying. Thank you very much for highlighting that.
Thank you.
Thank you very much, Dr. Rotimi. Yeah, I can see that. In fact, it's getting to 2 now. Somebody is asking, please, is there any online training to harness the opportunities in genomics and bioinformatics research? Is there any online training to harness the opportunities?
Yeah, so there is a lot of online training. So if you go on the H3A Bionet website, they actually have training, even like data sets, and there's a lot of training, that's available. So what you just need to do is just access, it's a public accessible website. So H3 Bionet is a project that came out of H3 Africa, which really was earmarked to enhance training. And also they do classes. So they have this training material, you can access even what they've trained before. You can actually access it, the tutorials. You can even download data sets from their website and even work through. They also do classes. So they also have a schedule which they advertise to even pace you through. Again, they even have videos. So yeah, really, we have all those resources. And even we are even pushing even further. They even like tutorials of even making pipelines. So fine, there are many other ways, tools that are there, but you can even package all your tools into one thing using pipelines. So all those things, it's available on the H3A Bionet website.
Thank you very much. Is that taken? Sorry, there's one more question, so we don't forget it. For an early researcher in genomics and bioinformatics study, where do I start from? I'm really keen about this. An early researcher in genomics and bioinformatics, where does the person start from? The person is keen about knowing where to start from as an early researcher in genomics and bioinformatics.
All right, so I think we also need to demystify, bioinformatics, because some people feel like, hey, it's really this amazing or very big or something that's difficult to attain to. But what you would start is just to learn a simple programming language. So there are many tutorials. You can start to learn R, just the basics, or even learn Bash scripting. Then if you are really keen even to start, it all depends on what level you are. Maybe you want to do a master's. I would even urge you, there are many people who are actually even looking for students, like the H3 Africa is a good portal. You can just apply, then you are enrolled, then you can even learn more as you are doing your master's research. Or maybe you are someone who's just keen to learn these skills so that you can apply maybe in industry. You can even go on those items. So we are now actually even having programs that are even like a master's in bioinformatics. We have such things in South Africa, or even masters in data science. Those are key aspects. Then even if you want to push further, you want to do your PhD, it's just getting a supervisor who's also in this space. People like myself, or even people like Professor Shegu, or many others. People are constantly looking for resources, people to mentor.
Thank you very much. Somebody is suggesting online that you can check Biotechnica for trainings. You can check Biotechnica or Omics Logic. Biotechnica or Omics Logic. Let's have your question.
Thank you, Dr. Tinashe, though I know you don't like to be called doctor, so thank you, Tinashe. So I just wanted to comment on the, you made mention of polygenic risk score. So I just want to comment on the transferability of polygenic risk score using other population to predict an African population. And here is my question. Like we all know that most other ancestry do not really predict well when we predict them using on African population. So my question is it possible to use other African population to predict within African population? For example, you're using population of Uganda to predict maybe Nigerian population. So I'd just like you to make some comments on that.
Yes. That's a good question. So what we're seeing in our studies, it's true that the European derived polygenic risk scores don't work well in Africa. What works better is like the multi-ancestry, which is a diverse, that also includes Africans, but also coming on the African continent, we've actually realized that scores that are derived, for instance, from Nigeria, they'll work in Ghana, but they'll work not so much in Kenya. So because of that regional diversity, but also there's also difference also in environments. So for instance, in the BMI paper that we are working right now, we realize that you can have a score that is created in Burkina Faso and Ghana. It doesn't perform very well because it's a rural area. Then you take that score and you predict in Soweto, this is an urban location, you see it is improved in prediction, but the genetics are a bit different. So there's just that combination of the environments being different and also the genetics being different. But for now, region-wise, if you have something in the East, it will work in the East, something in the West will actually work in the West, something in the South will work best in the South. But we just anticipate that this is just something that we can work around just for us to design better ways of actually correcting for the genetic differences and also the environmental differences that are at play. And this is a project maybe that someone might be keen to do, whereby you can optimize polygenic prediction. Because remember, in Africa, we have limited resources. So we don't have that luxury to say we have polygenic risk scores that work in West Africa. Like as I showed you on the map, our studies are still fewer. But we would need someone who can actually optimize this polygenic risk so that they can work in the whole of Africa using African data sets. And that is something that is of interest currently that we are exploring, but sure, we'll definitely find. It's just a computational and a statistical issue that just needs to be resolved. Thank you, Oka.
Very much.
Okay, thank you very much, Doctor. It's been so enlightening to see what you have done, and thank you very much for talking on leveraging bioinformatics and genomics. My own question is this. In your summary, I think the top point there, you mentioned on building and rededicating infrastructures. So I am a mentor with learn.omics.com, omicslogic.com, and I am Nigerian, I'm African, though based overseas, I want to see Nigeria prosper. Okay, so infrastructure-wise, our servers is in Israel. We get our resources from Tauba Bioinformatics Center. So from your own prediction, looking at the reality of our economy and without ignoring the power of political influence.
Yeah.
How will you predict that we accomplish your second summary point? That's my question.
Yes, no, thank you so much. That's a good question. And if you could see in that file, what started was political will or stakeholder will. They should need, so the essence is, let's say everyone really understands the value. Like, what does genomics give to us as Africans? As you are saying that if we understand the value, then if there's also that political will, that can happen. So I can give you, for example, in South Africa, I'm part of a study which is called AWIGEN. So there used to be demographic health surveillance sites. So these people used just to monitor health outcomes on the African continent. So when the HT Africa came, those same sites, because they only had phenotypic data, those same sites were now used even to genotype people. So meaning using that same infrastructure, which had been well coordinated, and the servers, okay, fortunate in South Africa, the servers are actually in South Africa. So they also established in South Africa. But remember, this was also as a result of strategic funding. Like H3 Africa also actually gave funding for that. So it can actually happen to say, for instance, in Nigeria, there can be centers, for instance, that have been pivotal for managing particular diseases. They've collected a lot of phenotypic data. They even have labs there. What then stops us just putting genomics as an underlayer? To say in this lab, you do this, can't you just, in the same time as you are looking for a certain disease biomarker, can't you then take the blood and genotype it? It's just adding a small equipment. Yes, you talk about servers. What about just getting strategic funding, which is just earmarked to make sure you actually have those servers in Nigeria? There's nothing that stops you from having servers. For instance, in South Africa, as a university on its own, we have 5 pentaterabytes of a server that is that capacity. This is just one university. So that can really be done. But as you rightfully say, it calls for that political will. And also it calls for academia, industry, making that advocacy to say, we need this. This is going to benefit us. Because just imagine you have this 100,000 Nigerian heritage study that is coming. How are you going to analyze it? Do you still need, things to be far away? Really, it means also the computational facility needs to be upgraded. People need to get funding. Government needs also to get funding, or even pharmaceutical companies need to be lobbied such that this can actually happen. But really, I mean, it's really possible. If it's being done on other African countries, why not in Nigeria?
Thank you very much, Dr. Tinashe. Somebody has requested online that you could share your email address so that they can reach out to you. Thank you very much. Are we done with this session? Then let's have Dr. Chinenye. Sorry. Let's clap for him. Sorry. We appreciate your presentation, Dr. Tinashe. Thank you very much.
Thank again, Dr. Tinashe. That was very energetic presentation. So we are now going into oral presentations. The first presenter, we're going to have 8 minutes for presentation and 2 minutes for question and answer. So we are calling on Richard Mayanja, sorry for wrong pronunciation. The topic is a meta-analysis of estimated glomerular filtration rates among African ancestry. Individual discovers PDZ-KI as a novel locus for EGFR ancestry. Please, we welcome Richard. He's online. Yeah.
Thank you so much.
Yeah.
I hope you can all see my screen. Good afternoon, everyone. I'm Mayanja Richard, a PhD student in bioinformatics from Makerere University. I'm here to present to you part of my research work that I've so far carried out and some of the findings from this work. And these are my co-authors. So in this work, we have used meta-analysis of estimated glomerular filtration rate to discover PDZ-K1 as a novel locus for eGFR of serum creatinine. Just to give you a small background, in our bodies, the kidneys filter blood, then they remove waste through urine. Therefore, if the kidneys don't function properly, wastes and toxins accumulate in the body, leading to a number of health complications. So unfortunately, 10% of the global population has got some form of kidney problems. And in Africa, the situation is even worse, whereby around 15% of the general population of adults living in sub-Saharan Africa have got some form of kidney problems. Previous studies, however, have shown individuals of African ancestry to have a 4 times increase the chances of developing chronic kidney disease compared to those of European ancestry. By chronic kidney disease, I mean a gradual loss in kidney function over a period of time. Unfortunately, not much is even known about what could be the genetic determinants of this loss in kidney function. And so far, the little we know is that the ApoL1 gene is associated with the increased risk of kidney function in sub-Saharan Africa. However, these studies have not been carried out within the African continent. They have been carried out among African Americans who are not even found within the African continent. And the only GWAS of kidney function in Africa was carried out by Fatmo et al. And here he discovered GAT-M and HBB to be associated with kidney function. However, the limitation with this study was that the sample size was not large enough. Therefore, the study was underpowered to reliably detect true genetic association within the diverse African continent. In order to try and address those problems or those challenges, we undertook this research study with the general aim of evaluating true genetic determinants of CKD in sub-Saharan Africa. So far, what has been done? To properly understand our workflow, please take note of these three important points. Please remember that the main measure of kidney function is estimated glomerular filtration rate. And this tells us how well the kidneys filter blood. Also, the second point, remember that the preferred biomarker for measuring eGFR is serum creatinine. And this is a waste product produced from the muscles. Thirdly, don't forget that a normal adult has got an eGFR of 90 mils per minute per 90 mils per minute per 1.7 meters squared and above. Therefore, any person found to have an eGFR less than 60 mils is considered to be having CKD, which is a gradual loss in kidney function. So in this research work, we analyzed summary statistics of the GWAS of eGFR of serum creatinine using 2 African cohorts. That is the UGR, the Uganda Genome Resource, having participants from Uganda, the African American Diabetes Mellitus Study, having participants from West African countries, which include Nigeria, Ghana, and the rest. So here we performed a meta-analysis using Neto, which is a command line tool to obtain SNPs with genome-wide significance, which is 10 to the power -8. We went ahead to do Bayesian fine mapping here to determine of the significant SNPs, which ones have true causality. Then we went further to do functional analysis and expression analysis. And finally, we performed Fewers analysis, which is a phenome-wide association analysis. So what did we find? In our meta-analysis, as represented here in the Manhattan plot and QQ plot, the Manhattan plot here is trying to give us a visual representation of our results, whereby on the y-axis, we have the negative log for the p-values. So meaning that our level of significance increases as we move upwards the y-axis. On the x-axis, we have the chromosomes. And here we can clearly see the location of the SNPs per chromosome. The blue line here represents the suggestive significance threshold. However, our red line here represents the genome-wide significance threshold. So this is what we considered. For all SNPs found above this red line, they are considered to be genetically significantly associated with kidney function. And as you can see on chromosome 1, we have a SNP there. We have on chromosome 5, we have on 11 and 15. And our QQ plot here was basically plotted to try and assess for the selection, if there is any selection bias within our study. So after finding our significant SNPs, we went ahead to see which ones are the lead SNPs. We found 4 of them which mapped to these genes. However, the SNP which mapped to NUDT1 and this other one which mapped to this 4th gene were left out. This is because they were intronic. And also the HBB gene, as earlier told you that it had been discovered by Fatum and the colleagues to be associated with kidney function, we left it out. So our study considered the PDZK gene is the one we followed up for our analysis. We carried out a tissue expression analysis for this gene, and here on this graph, the y-axis represent the transcripts per million, meaning the level of gene expression increases as you go upwards. Then on the x-axis are the genes in which this gene are the tissues, sorry, in which this gene is expressed. And as you can see here, our study showed that this gene is highly expressed in the kidney, and that is the cortex and the medulla. Finally, we carried out a phenome-wide association analysis. Here I wanted to see which traits are associated with this gene. And this table below here summarizes the traits which are significantly associated with our gene, this gene we discovered. So, although it was earlier found that this gene is associated with serum urate, which is also another biomarker for kidney function, our study also identified it. However, no study had identified this gene to be associated with kidney function, that is using serum creatinine as a biomarker. Therefore, in conclusion, for the first time, our study showed that PDK1 gene was significantly associated with estimated glomerular filtration rate of kidney function. We hope to carry out more analysis involving various cohorts within the African continent, among which is the Meru cohort. Here we shall get the genotyped data, perform GWAS, obtain the summary statistics, then from the obtained summary statistics, combine this with the rest of the cohorts within the African continent, then perform a meta-analysis from which we shall do computational analysis to perform drug target analysis. Before I conclude, I would like to thank the members of the African Computational Genomics group headed by Professor Shegun Fatumo and the rest of the colleagues, MRC, Uganda Virus Research Institute, London School, where I am a PhD fellow, Makerere University, and all of you for listening to my presentation. Thank you so much.
Richard, for your presentation. Please, do we have questions? Any question? Any comments? Any question, comments? In the absence of any question, we can proceed to the next person. Thank you, Richard. Thank you so much for your presentation.
Thank you very much, Dr. Chinenye. So we're having the next presentation, which is also online. We'll be having Adolaye Diawara. Hello. Adolaye Diawara. Our technical team, please help us bring him up.
Okay, hello.
Hello.
Yeah, I'm trying to share my screen. Okay. Hello?
Hello.
Hello?
Are you there?
Yes.
Please share your screen so that we can go on from there.
Okay. Okay, can you hear me? Hello?
Yes, we can hear you.
Okay. And my screen also?
Yes.
Okay. So my name is Abdullah Jawara, PhD student from African Center of Excellence in Bioinformatics of Bamako. And I'm going to present my work under the title Epidemiology and the Risk Factor for Type 2 Diabetes among Rural Residents in Sikasso, Mali. So, okay, I want you to retain this number. 463 million people worldwide are living with diabetes. This number means that if one has to create a country of diabetic people, such a country would be the third most populated country in the world. This number means that diabetes is the biggest pandemic in human story. So let's talk about the epidemiology of type 2 diabetes in Mali. The rates of diabetes in Africa is alarming. In 2015, people living with diabetes in Africa were 40.2 million. This number raises to 19 million in 2019. And the prediction is by 2045, there will be 47 million people living with diabetes in Africa, which is more than the double of the disease burden in 2019. Nowadays in Mali, even there is not yet a serious study to determine the prevalence in Mali. The type 2 diabetes constitutes a real public health problem with a huge impact on national budget. According to the Federation of Association Fighting Against Diabetes in Mali, 9 to 10% of Malians have diabetes. The challenge is that in the last 15 to 20 years, health system and research were focused on tackling infectious disease. The increasing in type 2 diabetes hasn't seen as a priority. This had led to several gaps in fighting against this pandemic, including a lack of knowledge about the disease among researchers. To shed more light on the epidemiological aspect of type 2 diabetes in rural areas of Mali, we set to ourselves the following objective. The first was to determine the prevalence and risk factor for type 2 diabetes in rural area of Niena municipality. And then the second was to analyze the risk factor related to type 2 diabetes. Sample used in this study were collected from Niena municipality located at 200 kilometers southeast of Bamako in Sikasso region, with 45 villages, mainly populated by Fulani ethnic group and Bambara, attracted mainly by agricultural and pastoral activities. You can see in yellow the map of Niena municipality. So here I'm showing the flow chart of my study. As you can see, our sample concern villagers over 20-year-old in Niena municipality. After questionnaire, anthropometric measurement and biochemical indexes to determine diabetes, pre-diabetes, and healthy participants, we perform the univariate analysis using as independent variables gender, age, occupation, hypertension, family story of diabetes, body mass index, waist circumference. And after that, risk factors with statistically significance in univariate analysis were used to perform the multivariate analysis. As a result, you can see in panel E, the prevalence of type 2 diabetes in overall participants was 7.8% and 8.9% with female and 5.6% with male. And for the pre-diabetes, the overall prevalence was 4.4% and for female was 4.1, and for male was 4.9. In the panel B, you can see it is commonly known that the prevalence of type 2 diabetes increases with age, which was noticeable in our study, but more in female than male. You can see also that the prevalence among participants in 40 to 49-year-old was approximately the same in male and female. Panel C, you can see the prevalence of pre-diabetes in female was significantly higher than in male in the same age. I mean 40 to 49-year-old. And you can see here the invariance analysis suggested that hypertension status and family story of type 2 were significantly associated with type 2 diabetes. The result of multivariate logistic regression analysis displayed that family story of diabetes and hypertension were independent risk factors for type 2 diabetes, and the difference were statistically significant. The take-home message is that the prevalence of type 2 diabetes among the rural population of Nyena municipality tested in this study was high. Hypertension and family story of diabetes in the first degree are strong predictors for developing the disease. A sizable genetic study is vital to validate the strong genetic association to type 2 diabetes found in this study. I cannot end this talk without my profound gratitude to my supervisors, Professor Mamadou Jakte, Dr. Talib Youssef from India, and then Professor Mamadou Wele and Jeffrey from Tulane University, and Professor Seydou Doumbia, the PI of WCP, the grant was supporting my PhD. And I would like also my profound gratitude to the organizer of this conference. Thank you.
Okay, thank you very much for your presentation. Please, I understand it's an epidemiological study. I would like to know how you arrive at your sample size, because looking at the samples, your sample size, the normal individuals, the number was so high compared to the pre-diabetics and the diabetics. Then secondly, I didn't get clearly about the prevalence of type 2 diabetes based on gender, because you classify both males and females. So I would like to also get that. Thank you.
Okay, let me stop to share my. Please, can you repeat your question?
Okay, I was saying it's an epidemiological study, and so I would like to understand how you arrive at your sample size calculation, because based on your slides, the number of diabetics and pre-diabetics are far way lower than the normal individuals. Then the second question is, I didn't get your conclusion about the prevalence of diabetes, type 2 diabetes based on gender, and what could be a reason behind the differences in the prevalence.
Okay, thank you for your question. First of all, our study is sample size was calculated according to the population side. It was a randomly collect. Yeah, we is not focused on only diabetes. It was randomly we ask people to come and to see the prevalence. What we have done is to know among the sample size we calculated what is the number of diabetes. Your second question was about you say you didn't get well the prevalence. The prevalence in this study for female was higher than male. It can be because we are going to investigate in the genetic study. For the genetic study, it is the previous study for that. But what we can say now is that it can be the macrosomal cases. We found a lot of macrosomal cases. Maybe also the lack of physical exercise, like in this part of our country, you can see the woman don't move a lot like man. What I can say for that now, I don't know if I answer your question, all your question.
Thank you very much. Any other question?
Hello, thank you for the presentation, sir. You didn't include the ethical approval in the study. Is it not necessary?
Yeah, according to the time I had for this presentation, if no, we have done some thing ethical, but it was not very significant for this study. That is why I didn't talk about. If no, we have done, we have checked the prevalence according to the different ethnic group in this area. As I said in my presentation, this area is mainly populated by Fulani ethnic group and Bambara. The two sides, but there is other ethnic group, but it's not very significant. But we didn't found anything linked to the ethnic group.
Thank you very much. Let's have your question.
Thank you. From your presentation, I realized that the study was carried among the Nina municipality in Sikazo, Mali. But going by the size of Chikazo, Nina is just 1 out of 42 communities in Sikazo. I think that should not represent the whole population in Sikazo. The Nina municipality should be reflected in the title.
Hello, did you
Yeah, I agree with you. But it is the first study like that. But for my PhD, I'm going to investigate in Yena municipality to know what is the gene involved in type 2 diabetes in this area. But I have to know about diabetes, what is the risk factors, what is the reason of this study. I agree with you, but we have to start with somewhere in Sikaso. Sikaso, as you say, is very, after Bamako, it is our capital, it is a very big area of Mali.
Thank you very much. We have 2 questions online and a comment. The first question says, what is responsible for female prevalence, sugar or cholesterol level?
I think I answered this question already. I say that maybe we noted several cases of macrosomal situation among female, but is also a risk factor for type 2. And then the lack of physical exercise because in our culture somewhere in this part of can see women don't move a lot like man. That is what I can say for this question.
All right, thank you very much. Another one says, is there any diet associated with the prevalence in female?
We didn't see that. We noted only that there is a big association between the hypertension and the prediction for, I want to say, a family story of diabetes.
Thank you very much. There's a suggestion that there is need for further study on prevalence in female in that community. And another question is, you want to make a comment on that?
Yeah, no, I say yes. I agree with you.
Another question says, is there any significant difference, the descriptive statistics related across your variables? Is there any significant difference in this descriptive statistics relevant across your variables?
No, I didn't get well the question. What I want to say.
All right. Thank you very much. Let's go to the next session. A round of applause for him.
Thank you. Dr. Abdullahi. The next person is Oluwatoyin Moreni Keji. Moreni. So the title is In Silico Analysis of Azadirachtin and its analogs on the dihydrofolate reductase of Plasmodium species. Oluwatoyin Sharon.
Good evening, all. My name is Oluwatoyin Moreni Keji, and I'm here to present on the topic titled In Silico Analysis of Azadirachtin and its analog on the dihydrofolate reductase. My slide, please. Thank you. These are my outlines. Malaria represents one of the most serious threats to human health worldwide, and about 229 million cases and 409,000 malaria-associated deaths occurs worldwide in 2019. This has an enormous impact on the mortality and morbidity, especially in sub-Saharan Africa, where Plasmodium falciparum accounts for approximately 90% of deaths recorded globally. Ensuring healthy lives and promoting well-being at all ages is essential for sustainable development goal. And currently, the world is facing a global health crisis, unlike others, due to COVID-19, which has increased human suffering and destabilized the global economic around the globe. Preventing and curing this parasite disease still depends predominantly on a small number of drugs whose efficacy is reduced by the emergence of drug-resistant parasitic populations. Azadirachtin has been reported to inhibit Plasmodium species, and the dihydrofolate enzymes has been shown to be reliable targets for anti-malaria drugs. Therefore, this study provides a more economical way of finding hits by screening Azadirachtin analogs through in silico method rather than real wet lab experiment. The aim of this study is to conduct in silico analysis of inhibitory potential of azadirachtin and analogs against the hydrofolate reductase in Plasmodium falciparum. The objectives are to predict the binding and drug likeness properties of azadirachtin and analogs on the hydrofolate reductase of Plasmodium species, to predict ADMET properties of azadirachtin and analog, and to compare inhibitory potential of azadirachtin derivative with two clinical drugs, that is pyrimethamine and proguanil. The methodology. Protein, that's the hydrofolate protein, was retrieved from Protein Data Bank and was being prepared using Discovery Studio and was saved in PDB format. The binding site of this protein was derived using CASP, which is an online software to determine the amino acid that could bind to the protein. Now, the reference molecule, which is azadirachtin, was used to build a library using Swiss similarity, and the ligands retrieved from Swiss similarity were then prepared. That is, the canonical smileys were retrieved from PubChem, which were prepared using ChemDraw. And this was subjected to docking using Pyrex. Drug likeness prediction was done using the Pinky's rule of 5, and pharmacokinetics prediction was done using Swiss similarity. Now, the results that was observed from this, we observed from table 1 that the first 6 analogs of azadirachtin have higher binding affinity than azadirachtin, which is the reference ligand, except for compound 7, which shows similar binding score as azadirachtin, that is -8.8. Now, the compound also satisfies Lipinski's rule of 5 for drug likeness prediction, except for the same compound which violated this rule. Now, table 2 shows good pharmacokinetic properties with which all molecules have higher gastrointestinal absorption, except for the same compound which did not obey that. And 4 of the compounds possess blood-brain barrier permeability, and only 2 out of all shows the ability to transport drug permeability glycoprotein. Even though the compound did not follow suit with others, that does not mean this compound cannot be worked on. It's just an opening to tell us that further research can also be carried out to know what could be responsible for this. Now, in conclusion, based on the binding affinity or the binding score and the drug likeness ADMET properties, it is observed that the analog have potential inhibitory property. And this analog could also be used to synthesize for oral formulation as anti-malaria with further modifications. I acknowledge African Center of Excellence in Phytomedicine and Research Development, University of Jos. And I appreciate the Nigerian Bioinformatics Network for giving me this opportunity to come and present. And thanks to 54G for their support throughout this day. Thank you. For the people on site and online, I say thank you.
Thank you, Oluwatoyin. Thank you for your presentation. Please, do we have any questions, comments, contributions? Okay. Where's the mic?
Hi. I have a question.
Okay, online, please. You can.
Yes, Mamadou Sangare from Mali, African Center of Excellence in Bioinformatics. I would like to know, how did you get your target?
I got my target from literature search.
From?
From literature search.
Okay.
Thank you.
Thank you for your presentation. I believe we are talking about Azadirachta indica, which is neem plant. So you were supposed to tell us about neem plant before telling us malaria, because all that you've said here is all about malaria, malaria, malaria.
I'm sorry about that. But the interest here is on azadirachtin, which is the active component that is responsible for the anti-malaria activities in neem plants. So azadirachtin is just a compound from neem plants. Thank you.
Okay, thank you for your presentation. Yes, I have a small worry with regards to the plant, because dihydrofolate reductase, I think it's a commonly called Fancida, an intermittent preventive treatment for malaria in pregnancy. So currently there is an emergence and widespread of resistance. So if at the end of this study, we have this compound to be successful, how long might it take for it to experience the same problem based on the fact that it's an analog? So the same effect might be executed on it too.
Now, our research scope is not covering the resistance. What we wanted to know for now, if actually it could work or not. And also, this compound has not yet been synthesized. So until we have full information about it and we go for wet lab to carry it out, then that could be put in consideration. Thank you.
Good evening, everybody. Please, not a question, but it's more like a suggestion. And I think I discussed with the other guy that worked on ECG, EGFR in cancer. So as a rule, I think whenever we're doing all this kind of in silico study on plants, it's necessary for us to do, apart from the gastrointestinal absorption, blood-brain barrier, it's important to do the synthetic accessibility, because at the end of the day, it worked on the computer. But if you now want to translate it to real life, the first bottleneck you may have is this product or this compound cannot be produced in the laboratory, and that makes the whole work not. So I think you should also include it. There's a way you can do it in silico, but I don't know the database you use to run the analysis, but you should also, I think anybody that wants to do such a research should also put that at the back of the mind, that they should also check for the synthetic accessibility of the products or the hits that you've gotten from your analysis. Thank you.
Thank you.
I have a question online. How was the extraction done? What's the workflow?
Okay, the data used for this study are all secondary data. They are already deposited in the data bank, so we didn't synthesize anything, even though azadirachtin is already in the market being synthesized, and there is a company selling the already made azadirachtin, but we did not synthesize anything.
Thank you so much. A round of applause. Another round of applause for all of us. It's been a long day. Thank you all for your patience. I'm handing over to Dr. Itunu.
Let's put our hands together for the session chairs. We appreciate Dr. Ezebialu and we appreciate Dr. Ladelu. Thank you for rising up to the challenge. So we have come to the end, and I thank you all for staying up till now. Just a few announcements. So the cocktail and the closing ceremony is by 7 p.m. I think it's best we just hang around. The university management will not have us go, just like others have gone. They want to host us to a cocktail dinner at the Hall 3 at the cafeteria where we have been eating. It's just across the road. So during that closing ceremony, the conference is going to be declared officially closed. We'll have a small handover ceremony from the old GC to the new GC. We've already done something online, but just to situate things properly. And we have plans to also announce the winners of the poster and the oral presentations. Right now, I'm collating results from the reviewers, and we are trying to do that. So 7 p.m. prompt, we will be meeting at the hall 3 of the cafeteria. And then there was something on the program about plans for the next conference. I don't know, maybe when we have more information, we can just talk briefly about what we want to do, but the plans are still not concrete yet. So that's it from me. Let me invite the president to just close us, and then we'll have a proper closing. Question? Okay. I just came to close us, so not to answer questions.
Okay, I just want to make a request. That is it possible for you guys to share the conference?
I have emailed every one of them.
Okay.
They have received my email. So no, the people that the keynotes, you mean?
Yes.
Yes, I have emailed the keynote speakers. That's what I'm saying. I've emailed the keynote speakers today. Dr. Rotimi can check his email. He will find my email there. Okay. Yeah. So while they were presenting, I emailed all of them and I said, oh, thank you for your wonderful presentation. Please send me your slides. As soon as I do have it, I will push it to everyone. But I don't want to be doing it in bits and pieces. I have the ones from yesterday. I want to compile the ones from today. And once it's complete, in fact, I don't even think I will email it. I will put it on a Google Drive and send the link so that everyone can access it. But I hope nobody goes there to delete it. That's another thing. I've done that before, and I've done it before, and somebody went there and deleted, mistakenly maybe, deleted a file.
What about the slide that Dr. Fatima made? Is that one also available?
Dr. Fatima's what?
Slide presentation.
Did he make a slide? It was a welcome speech.
And it was in the form of a slide presentation, man.
To who? I don't have it. Send it to me and I can put it up for everyone. Please, if you have any comments, maybe I should allow you to take comments. If I can answer them, I'll try to answer. But I have plans to send everyone as soon as I get it complete. I won't disappoint you on that one. Therefore, the certificates, people have been asking me, and I've said that we are going to do e-copies. The LOC is on top of the situation. I've sent a list of all the people who register, whether they are here or not, to send e-copies to your emails. So there are some people that will not be on that list. If I don't have your email, if you did not officially register them, I might not be able to send. So you might need to do an email to me so that I can copy that email and send it to them to do it. So if by the end of this week you don't see it, then contact me or contact the LOC. They are the ones handling it. Dr. Baje is the deputy co-chair of the LOC. He's the one managing that for me. Then for the travel fellowships, I said before that the partial fellowships do not cover transportation, unfortunately. But those that are on full fellowships, I've sent you a Google form. Don't be deceived. I will go through that Google form again. I'm already seeing double entries. I am a manageable girl. Let me remind you. So I will not make mistake to pay you twice. I'm assuring you of that one. So I'm going to look very well before transferring money. And I will make sure that by the grace of God, I do not transfer money twice. So, but if you receive it twice, I know you are a good citizen. I know you're a good citizen and you'll be very happy to send it back to me. Okay, but I'm going to try to do due diligence and not send it 2 times. So, yes, question.
For membership NBGN, can anyone?
Membership certificates.
Yeah.
We don't have membership certificates.
Okay.
We don't have them. Why do you need it? Okay. Have a membership number, right?
Yes.
Okay. Okay, good. President, sir, this is for your attention. When you come up, you will address it. I don't have answer, and I don't want to give you a wrong answer. So the president will address that. It's beyond my office. Let me not go big. Let me do you have any other questions to address? So for the town fellowship, we will not be giving cash. There's only one person that is not in Nigeria is not in Nigerian that will handle that one separately. But for Nigerians, we want to be compliant with the cashless policy. We are law-abiding citizens in NBGN. So we'll try to do the transfers, but I am not promising today anymore because it's already late. But first thing tomorrow morning, we'll take the schedule to the bank as soon as I've confirmed that there are no double entries. That was why I accepted your bank details by form so that I don't have to be copying and typing one by one. So I think that's it for me. I've enjoyed myself. It was beautiful hearing from all of you. I was not disappointed by the quality of the presentations and much more the engagement. I think I even enjoyed the question and answer sessions. Do you agree? People really asked very exciting questions, and people also provided solutions or possible ideas. And so whenever we present like this, it's not really about even the questions. I want to even get more feedback from people, and it will certainly give you more ideas to how you can expand your work and extend your work. So we are hoping that in the 2 years' time, when we call you again, we'll have a bigger house. Unfortunately, this year we had a lot of last-minute planning and all that. We're very sorry for it. The pandemic really didn't make us decide on time if we wanted to do virtual or wanted to do physical. We're confused at some point. But I want to believe that it was not a bad outing. Do you agree? Yes, at the short notice, it wasn't bad. And I really, 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 really enjoyed the keynote speeches. I enjoyed the invited speakers. We didn't make any mistake by bringing any one of them on board. It was really beautiful. And thank you to all of you for making a very exciting time. Many of us are tired, I must tell you. It was very, very neck-wrenching to plan this conference. But I don't regret it one bit. I've put a lot of things aside just to be able to do this. And I'm happy that by the grace of God, it was a success. So let me stop talking because I can talk to tomorrow. Let me stop talking, and I have the singular privilege to bring the chairman, the president, NBGN on board. Please put your hands together for Dr. Charles Adetunji. Thank you, sir.
NBGN, you will say, what should we use so that, no, I think we should have a slogan. Am I right? Because, okay, so what should be our, when we say NBGN, let's decide that now within 1 minute. Huh? Okay, Prof, you said. Okay, Doctor, what do you want to say? Eh? This is not a code.
Hello, can you hear me?
I want us to have something that we stand on, and we are looking beyond where we are today. We are
I want to give a suggestion.
So NBGN.
For the greater good.
Okay, maybe we talk.
For the greater good.
Okay. Okay, I like that. Projecting into the. Okay, do we accept that? Okay, NBGN. Okay, we do what? Okay, let's think about it. Maybe during when we want to have our meeting.
I want to suggest.
Okay.
That it should.
Okay. So, sincerely speaking, without wasting time, I want to thank everyone. I want to thank our distinguished Professor Rotimi, a highly classed professor of his kind. Please, let's clap for him. Let's celebrate him. And I want to thank all the professors in the house. I can't count as many as possible. Distinguished doctor, distinguished students, and distinguished researchers and erudite scholar. It's something that I see us as a kind of body that we are going to solve the global problem. I see us that the whole world will be looking up to us in some years to come. Now, By the grace of God, without wasting time, I want to congratulate everyone. But before going, please talk to your neighbor. Just say congratulations for making it. Please, let's smile and talk to our neighbor. Congratulations for making it. Wow. Wow, I love this. Please, some people, they are not doing this, please. I will still shake everyone, no problem. So we shall be moving on to the cocktail party. And before doing that, I want to assure everyone that we have everybody have something to take home. Two of us, no matter wherever you are before, you are in another level. And I know as you are stepping out, you are going higher. Having said that, by the grace of God, we are going to consider all the paper in a high impact factor paper, index in Scopus or Thomson. Not only that, we are going to make call after that. And there are a lot of book opportunity, and Professor Fatima are editing currently because of recent I was appointed as a series editor in US by Francis and Taylor. So we'll talk about that very soon. So we have more than 10,000 books in that series. And I am Professor Fatima will be coming on with the first book of that kind very soon. So I will throw it open to the whole house if you have chapter or you have so many things to key into. Not only that, about certificates, my vice president said I should address that, and on behalf of the board, we will decide on that. And definitely, I think it's very necessary for us to have an evidence so that we can beat our chest. I belong to this house. Let's clap for ourselves. So without wasting time, we have a meeting properly so that we not miss the chicken, the turkey, the haruma, and the flavor, and the drinks. So maybe we go together. But there's one thing we must not miss. Make sure you connect with nothing less than 30 people before you go home. Hello? Make sure you connect as far as possible. So, by the grace of God, I want to return all glory back to God. Whatever we might be, we are just penciling the hand of Almighty God. We have seen, we have traveled, we have conquered. And I believe by the grace of God, we'll return back home safely. We meet our children, we meet our wife, we meet our lovely ones back by the grace of God. So as we go now, we go and march straight into the cocktail party. God bless you and remain blessed. On behalf of, I want all of us to celebrate the Fissionaire. Okay, please help me celebrate my vice president. I think she's going to be a very distinguished vice chancellor very soon. And she's even taking the seat. So she said something that I'm giving you the microphone to echo it out.
Okay, so on the logo, we already have for the greater good on the NBGN logo. So if you think that that's okay, we could use it. Or we could also consider something else. But on the logo that we inherited, it wasn't us that designed it. It was the original GC that designed it. So if you look at the NBGN logo, you will see for the greater good on the NBGN logo. You can check it. So good.
I want us to be in the curry party before the vice chancellor will march to the venue. God bless you and remain blessed. I take a bow.
Please make sure you return your bed sheets while you are departing. The bed sheet and the keys to your room should be returned.
Before we go, help me celebrate the chairman, Professor Marion,